Dr. Avindra Nath, who is the intramural clinical director at the NINDS at the NIH, and he's also chief of the section of infections of the nervous system, and has had a huge impact in our thinking about the role of retroviruses, in particular HERV in ALS, with some extraordinary experimental findings in animal models of supporting that view. So Avi, thank you so much for joining us. I turn it over to you now.
A very kind introduction and for inviting me to this meeting. I really enjoyed all the talks. So let me see. I hope you're able to see my screen now.
Yes.
Okay, so I hope we're in presenter mode here. Okay, so it's always a tough act to follow Josh. He does such a beautiful work and really beautiful concepts. I'm going to show you at least where we stand in part of the discovery of the endogenous retroviruses in the context of ALS. And so I've divided my talk into 4 parts. First of all, what are endogenous retroviruses and how did we acquire them? The second is, do they serve a purpose? Then third is, what happens when they get dysregulated in the brain? And that's where I'm going to spend the majority of my time on this talk. Then lastly, I'm going to tell you a little bit about can we actually control them? So this was the discussion that we were just having, and that is just as you said, Bob, our protein coding genes are only 3%, and the rest of it has all been acquired through some retroviral gene, and certainly 8% of them are really endogenous retroviruses. So that's almost 3 times what the protein coding region actually is. So when you do whole genome sequencing, what people are really studying is only this 3%. Okay? And so when they say whole, I really call it whole like this. That's what you're studying when you do whole genome sequencing. Really all this data gets thrown out. And it's impossible for me to believe that this part of the rest of the DNA doesn't play any role in all the genetic diseases or whatever illnesses we study. And we keep talking about ALS here, we got this mutation and that mutation, we find it, don't find anything else because you never looked for it and you threw out all that data. That's why you don't find it. Yeah? It's all sitting in the rest of the DNA here. So anyhow, the way we acquired them, if you look at our non-human primates, which are our closest cousins, over a period of time, we acquired a lot of these endogenous retroviruses. So they keep getting inserted. And our closest cousin here is the chimpanzee. And what separates us from a chimp is the presence of the HERV-K. Yeah? And I remember a talk that Josh once gave, and it always struck in my mind. And that is that what he said was that if you look at a mouse and a human, 99% of our protein coding is exactly the same. But yet a mouse is not a human, right? And the reason it's not a human is because the endogenous retroviruses that the mouse has are very different than what humans have. So we split at some point in time and we started acquiring different types of viruses, and that made us into different species. And Josh, if I could quote you even further, he said, I remember you once said that if you look at the protein expression part, there's 50% homology between a banana and a human. Is that right? So if that really is true, then really it's the rest of it that's more important to study than the protein coding region. So this is how we acquired. The interesting fact is, if you look at all retroviruses, every retrovirus is sexually transmitted. So you ask yourself, why is that the case? Why would a retrovirus want to be sexually transmitted? The only reason it is it's trying to get into the germline. So if you have a retrovirus gets into the germline, retro because it goes from RNA to DNA. If it can go from RNA to DNA, it gets inserted into the chromosome. And then the next offspring now has equivalent of, let's say, 10 kilobases of new DNA. And this has everything in it. I mean, it's got promoters, enhancers, enzymes, structural proteins. And as just Josh showed, it can cut and paste itself throughout the genome. So it can do good and bad things depending on where it lands, and multiple copies can actually form. So most evolution occurs this way. I mean, once we became humans, we haven't actually evolved. We are no smarter than our ancestors that lived in the cave. right? They just didn't have access to cars and cell phones and the like, right? But so humans have not evolved in a long time. But what has evolved, if you also look at, is the brain. Okay, so as we evolved through the non-human primates to humans, the size of our brain and the complexity of our brain also changed. So a lot of the retroviruses are important in brain function. So what purpose do they actually serve? So all mammals have placenta. And if you have placenta, the placenta is made of the envelope of a retrovirus. And so that's what they form syncytia. That's what envelope proteins like to do. So and for each mammal, there may be a different endogenous retrovirus that does it. For humans, it's called HERV-W that forms the syncytia. And that's what forms the human placenta. So I'm going to focus more on HERV-K. And so if you look at this endogenous retrovirus, which is the most recent one we acquired, and when you're talking about recent, you're still talking about a few 100,000 to 1 million years ago. And in the human genome, there are 140 HERV-K elements, and they form 330 kilobases of the human genome. And they're classified based on the reverse transcriptase. So the HERV-K is not a single virus. If there are 140 elements, that's equivalent of 140 viruses. And you can actually group them into 11 different subtypes, and they're named as HML1 through HML11. And here you can see that there are all these, in every chromosome, you can find HERV-K elements and multiple copies in each of the chromosomes. Okay, then if you further classify HML2, what you will find is that there are about 89 insertions in the human chromosome. 23 of them are transcriptionally active, right? And 17 are polymorphic. These polymorphic ones haven't actually been studied very well. So if they're polymorphic in the human genome, they could do all kinds of things. Based on the envelope gene, you can classify them into type 1 and type 2. So 74% of these are called type 2, and that is it has the complete envelope, and it will form the so-called REC protein, which is spliced off the envelope. There can be a 292 base pair deletion, which will lead to what is called type 1. If you have type 1, then it forms this other protein called NP9. So all HML2 is not exactly the same. There are multiple different types of HML2s. And then based on the LTR sequences, you can further classify them into 3 different subtypes. And there's 5 HS for human specific, which is the most recent. Okay, so we look to see that can you actually find them? Do they serve a good purpose? And can you actually find them in through the process of human development? So we looked at pluripotent stem cells, and here we stained it with the envelope protein of HERV-K, and you can find humongous amounts of expression, huge amounts of expression. And they're absolutely critical in keeping these stem cells together. If you block it, these things will fall apart. But if you move them from a stem cell, a pluripotent stem cell, to a neural stem cell, and then to a neuron, it will get silenced. And this silencing is extremely important, because if it doesn't silence, you're not going to form neurons. Okay. And if you force the silencing through a siRNA, you will see that they will differentiate to neurons immediately. And as you can see, as we force the expression, you can see nestin expression, showing you that it's actually moving along the neuronal pathways. So we look to see, okay, where is this envelope coming from in the human genome? Because I told you there's so multiple copies. And we found that there's a site on chromosome 19 that is capable of producing the entire full-length envelope. And that's where most of it is coming from. Although the transcripts from various other places, they're not able to form the complete protein. And we found that sure enough, at this site, as you go from a pluripotent stem cell to a neuron, it gets more and more methylated, so it gets silenced through the process of epigenetic changes. We then mapped out the entire pathway as to how it maintains the stem cell. And since it's published, so I'm not going to go into details. But however, to say that we identified a receptor, which is the CD98 heavy chain. And that signaling through that heavy chain is absolutely critical. This binds to the alpha-1 integrin, and it signals via that mechanism. Okay, so what happens when it gets dysregulated? So Josh already mentioned that these things can be implicated in a number of different diseases, and here's one showing that at least a number of neurological diseases you can implicate these endogenous retroviruses. They've been very poorly studied, so people really haven't studied these things in various diseases yet. HERV-W has been studied to a fair degree in multiple sclerosis, and I'm going to show you data on HERV-K here. So the idea that retroviruses are associated with motor neuron disease is not new. Okay? So there's an interesting story here from NCI, and what they were in the 40s and 50s were interested in. They said, we need to find out these RNA tumor viruses and DNA tumor viruses. That's how they classified tumors. And if you can find them, then what you can do is you can immunize people and you'll prevent all these tumors from occurring. Okay? So that was the concept at that time. So this guy, Murray Gardner, was tasked to try and find these RNA tumor viruses. So he was at UCLA and then later moved to UC Davis. And what he did was he would go to these squab farms. These are like pigeon farms in LA, in Ventura County, and then duck farms and grain mills in LA County. So these pigeons were important for all the, in Chinatown for all the food products. And so ducks were also eaten, so was grain. So they had to be rodent free, okay? But instead what he did, he said he went out there and he would catch all these mice. And he was trying to find out if they have a virus that he could isolate. So he wanted to find them in the wild. And then he was trying to isolate and see if they would develop tumors. What he instead found was that there was this one set of mice. He found at 3 different farms that they were developing this lower motor neuron disease. Okay. And it would occur spontaneously from this virus. And he isolated the virus and he called it a type C RNA virus, which is nothing else but a retrovirus. Then there's a whole body of literature for 20 years, of extensive body of literature trying to determine how does it cause lower motor neuron disease. And what they found was that the envelope is absolutely critical in mediating pathogenesis. And the envelope alone can actually do it. It is transmitted via breast milk and sexually transmitted. There are sporadic B-cell lymphomas that occur in these mice. Has a long latent period. This is extremely important. A mice lives for 2 years. Latent period is 18 months. And when it infects the anterior horn cells, the brainstem and the cerebellum, there's no inflammation. Absolutely no inflammation. Okay. And then I'll talk a little bit more about HIV that can also cause an ALS-like syndrome. And HTLV1 is a human retrovirus that has been associated with tropical spastic paraparesis. Tropical spastic paraparesis is nothing else but a motor neuron disease. And some of these patients will develop lower motor neuron symptoms as well. So then they satisfy the criteria for ALS because you can get both upper and lower motor involved. When that original paper was published by Murray Gardner, Jacob Brody got very interested about it because he'd been interested in the transmissibility of ALS, like can ALS be transmitted or not, okay? And so what he did was he had access to these Guam ALS patients, and he did this very elegant experiment and very clearly demonstrated what he at that time called RNA-instructed DNA polymerase, that there was this humongous amount of reverse transcriptase activity in the brains of these Guam ALS patients. Okay. And now if you look at it, after that, a number of people have tried to look for retroviruses. Most of people have concentrated on trying to find exogenous retroviruses, okay? So there were a series of papers published showing antibodies to spuma retroviruses, and then a group claimed that they couldn't reproduce it. Then there was HTLV tax Rex was isolated. And then another group said, well, we can replicate it. And then a number of papers were published showing that reverse transcriptase could be identified, but they couldn't find the source of the reverse transcriptase. But these are elegant work done, largely coming from Amar Al-Chalabi and Jeremy Garson's group. I think Bob, you, and Merit have also contributed to some of this elegant work here. And then we published that we could find reverse transcriptase activity in ALS patients coming from HERV-K. And later we've identified as being HML2. So the classification of HERV-K has also evolved over a period of time. But I think it's fair to say that all that we were publishing is from HML2. And then there are two papers, again, one from Jeremy Garson, another from a German group claiming that they find it in ALS, but it's no different than controls. Okay. And then there's a whole body of literature. I'm going to go into this in a lot more detail, implicating HML2 in ALS. And then Amar just published a very recent paper claiming that he finds HML6 activated in ALS brains, but its pathophysiological significance is unclear. And Josh already showed the slide from work that he and Molly Hamill had done. So there's a subgroup of patients with ALS where you can see transposable elements activated. So Tara in my lab, she looked at a much larger group of patients from the same New York Genome Center database, and she classified them into 4 groups. Okay. The ALS was divided into 3 groups, and this is all based on expression of HERV-K genes. And so there's one group of patients that have absolutely no activation of HERV-K. There's another group that has only very few loci that are activated, or 5 loci. And then there's another group of patients that have 35 loci. Almost all of them are activated, right? But they are much greater than controls. So none of the loci in controls were always expressed more highly than all ALS patients put together. And here I'm showing you 3 different loci. So in chromosome 22, you can see this locus here. Each dot represents a single individual. And here are 2 different loci in chromosome 19. Right? Very clearly, you can see that the amount of elevation in the ALS is much greater than that of controls. And this is to show you, previously we've shown that we can detect a reverse transcriptase protein in the neurons of ALS patients. And we can also find the envelope protein here expressed in pyramidal neurons within the cortex, as well as the anterior horn cells, but not in the lateral horn, not in Alzheimer's, and other types of control tissues. We've also looked at Guam ALS patients. Through Daniel Pearl, we were able to get a couple Guam ALS patients. And we found that, yes, you can find activity of HML2 in various regions within the brain. But by Western blot, we could actually demonstrate full-length envelope that was present in the cerebellum of one of these individuals. Okay. And maybe a smaller amount in the second one too. Then we further mapped it, and we found that it was coming from chromosome 19, Q11. Okay, so if this is being expressed, so what? I mean, does it really do anything, right? Expression alone doesn't mean anything. So what we did was we transfected neurons, and with the HERV-K envelope and with the entire HERV-K, and what we found was that it is toxic to neurons. Okay. If it's expressed in neurons, it'll kill it. And that makes sense. I showed you earlier that what happens to HERV-K is that it's expressed in early embryogenesis, then gets shut down in neurons. It's being shut down for a particular reason, because if you re-express it, it does bad things. And we also used a CRISPR-Cas9 system to activate the endogenous HERV-K, and it did the exact same thing. Then we made a transgenic mouse model, and we found that 50% of these, the transgenic mouse model was made with just the envelope put under a neuronal promoter, and it forms full-length envelope protein here. And we found that 50% of these mice will die by about 9 months of age. They develop classical upper and lower motor neuron signs. Okay. And you can see here that there's fiber type grouping of the muscle fibers in the transgenic mice. You'll see that their movement is impaired. And if you look at the upper motor neurons here, you'll see there's dilatation in the neurites and loss of neurites as well. So there's impaired motor task, but the sensory vestibular system and cerebellar system is actually spared. If you look at the spinal cord here, you'll see that there are very few anterior horn cells left. The ones that are still there are actually much, much larger in size because they have to do the job of all the other anterior horn cells that are gone. Right? Then if you look at these neurons much later in time, so at about 11 months of age, you will find that TDP43 is now present in the cytoplasm. So it translocates from the nucleus, as you can see on the nucleus, to the cytoplasm. So it takes a while. You have to express HERV-K first, then you get translocation of TDP43. It's not the other way around. Yeah. And then Renee Duval in Canada, what she did is she looked at the HERV-K envelope and she found that there's an interesting splice protein that occurs, transcript that occurs off the surface unit. And this is spliced transcript forms a protein that is a conotoxin-like protein. And she found it at autopsy in the neurons of ALS patients in the brain, as well as in the spinal cord. So then Hervé Piron in Lyon, France, he got some CSF samples from us. And these are from 14 ALS patients. 2 of them are from a single patient here. And he looked for the protein, the envelope protein itself, for HML2. And he finds that he can find the envelope protein here. And this is just the surface unit because it's smaller in size than the full length. But he found it in several of these patients over here. Then Joe Steiner in my lab took the protein, put it on neurons in culture, and then recorded off of them. And you can show that the firing rate of these neurons actually goes down. These are 3 ALS CSF samples, 3, 5, and 9. And when he took an antibody to HERV-K, this is a monoclonal antibody, he could block it, but not with a control antibody for both of these individuals, 3 and 9. Here he shows with recombinant protein. So I made the recombinant surface protein of HML2 and put it on neurons. And in a time responsive manner, it causes toxicity. And if you heat the protein, you lose the toxicity. And you can also block it with the same monoclonal antibody to HERV-K. You can block the neurotoxicity. So again, now we've mapped the pathway in great detail. I'm not going to go into the data for all of this, but suffice to say that, again, we found even in these neurons, it's the envelope protein binds to the heavy chain of CD98 and then signals through the beta-1 integrin pathway as well as through the light chain and blocks the transport of cysteine that then causes oxidative stress. So next, a group from Italy looked to see if they can find antibodies to HERV-K in patients with ALS. And what they showed was that there's a particular small linear epitope, small peptide. So they mapped a lot of different peptides, but this was one particular peptide to which the antibody levels were extremely high in ALS patients compared to multiple sclerosis or Alzheimer's disease. And they found it both in serum as well as CSF, right? And then when they took this peptide and put it on B cells, on PBMCs, they found that it was stimulating B cells. So clearly these B cells have seen this peptide before, okay? So it actually is really elegant work. Now what I'm going to show you is they found that there were elevated levels of antibodies. What we did was we took the entire HERV-K protein there, and then we looked for antibodies. And what we found was that if you look at the HERV-K levels, as they go up, the antibody levels go down. So there's an inverse relationship between the two, okay? And if you look at the, yeah, so this is HERV-K itself. And over a period of time, the HERV-K levels themselves go up with duration of disease, okay? So this to us is interesting because here we're looking at the entire protein. So if you get antibodies against the entire protein versus a linear epitope, that means that there is most likely explanation of that is that there is epitope spreading. Okay. And that's why they were able to find it against a single linear epitope. That probably is not exposed till much later in the course of the illness. Okay, so now I'm going to say that how can we control it? So the first question you would ask that, okay, it's a retrovirus. Why don't we just give people retroviral drugs and we'll fix the problem, right? And so, in fact, that's how I got involved in ALS was because my expertise has really been in HIV for several decades. And stumbled across a patient who had HIV and ALS. We treated the HIV and the ALS got better. And when you looked at the literature, we weren't the first people. There were a number of reports in the literature saying that some of these patients develop an ALS-like syndrome and it's reversible. It's reversible in about 50% of these patients, but you have to give it early in the course of the illness. That tells you that early on in ALS, you don't have neuronal death. What you have is neuronal dysfunction or neuronal injury. And it is possible to reverse it before these neurons die. And these were a total of 5 patients that we had seen at that time. Now we've seen several others. And this is their ALS rating scale here. And you can see that if he started the antiretroviral therapy early on, within 6 months, this patient reversed completely. Here we started again quite early. The patient got better. Again, 6 to 8 months got better. You start here much, much later in the course of the disease, and you may have prolonged the survival, but didn't get better. Again, here, we started antiretroviral therapy, changed the antiretroviral therapy. We didn't alter the course. Okay. And we may have slowed it down at the best. We don't really know. But these patients survived for several years. We screened a whole host of antiretroviral drugs against HERV-K to see how effective they are and compared it against HIV. To our surprise, we found that abecavir was actually better against HML2 compared to HIV. All the rest of the drugs, they have some activity, but they're not that great. I mean, AZT, you'd have to give 3 times the amount. Darunavir, you probably have to give 25 times the amount that you would give to an HIV patient. So they have some effect, but they're not that great. Yeah. So at the time when we published it, Julian Gold actually showed up in my office. He says, I'm in London and I'm going to Australia, but I want to stop by and visit you here in Washington, DC. I said, okay, that's interesting. And then he tried to convince me. He says that it's absolutely unethical not to treat ALS patients with antiretroviral drugs. I said, it is unethical to treat them because I've been around long enough to know that despite all your best intentions, you can end up hurting people unless you do it in the context of a clinical trial. And so he did an open label clinical trial and he treated patients with Triumeg that had abecavir, which is what I told him, that this is the best drug if you want to use it. And what I told him was that, okay, if you're going to do the trial, make sure you save all the samples for me to analyze, okay, and which he absolutely did. And so we got the samples and Marta in my lab then measured the HERV-K levels. And interestingly, what we found was that over a period of 24 weeks, actually the levels did go down, but it never came down completely. You brought it down some. But what is most interesting is that once these patients went off the antiretroviral drugs, the levels bounced back. Okay, that clearly tells you that the drugs were acting on HML2, right? The other fascinating thing is this is an open label study, so take it for what it's worth. The sample size is really small, so it's subject to all the caveats. But nonetheless, we divided the patients into 2 groups. One we call responders, and the responders are those individuals whose viral load decreased. Okay, that's what we're calling a responder. It's not a clinical responder. This is the one that the viral load actually is, a virological responder. And a non-responder where there was no change or an increase in HML2. And just go to this right graph over here. When you now look at them, those people who are non-responders, you can see their ALS rating scale is coming down, except for 3 individuals here, their plummets. Those individuals who are responders, except for 1, most of them progressed very little. Okay. So I think this begs that we do a proper placebo-controlled, double-blind study to refute or confirm these observations. Yeah. The caveat here, of course, is that if the envelope alone is sufficient to cause toxicity, then the antiretroviral drugs are not going to work, because you can just you can have an open reading frame for envelope, you keep producing the envelope, will cause toxicity. Okay. And the other thing is we need to develop better antiretroviral drugs against HML2. So what we did was we screened a whole host of antisense oligonucleotides, and we cloned all these different loci of HERV-K, of HML2, and then said in culture, can we block it with our antisense oligonucleotide? So we identified one of them that blocks all these sites really well. And probably not that great for chromosome 6, but all the others, it blocked really well. So I think this is a potential therapeutic candidate that can be taken forwards. We've also developed shRNA, and we put it in an AAV vector, and then we injected it into these mice, and these are our transgenic mice with the HERV-K in here to see if that will block it or not. So we first just put GFP in these AAV2, and you can see beautiful uptake in the cortical neurons, and it goes down to lumbar 5. And here with the shRNA, and you can see that actually they gain weight just as well as the wild type. And if you look at the muscle here, you can see that this is the transgenic. You can see a lot of fiber atrophy, and here's protection of the muscle fibers here. Okay. So we're injecting it into the brain, and we're showing you here the protection all the way down to the muscle. And we quantified the number of HERV-K expressing cells, and they were decreased in the animals that had the shRNA. We now screened all the protease inhibitors, and we found that Durinavir is probably the most active, but yet it's 200-fold less potent compared to HIV protease. Okay. So what we've done is we modeled it. We've now made all kinds of analogs of Durinavir. We screened them and identified one that fit into this pocket a lot better compared to, and you can see that the modeling of this actually looks pretty similar. So it's quite possible to fit a drug, modify it, and make it fit a little bit better into this little space here. So in summary, what I like to say is that retroviral elements were acquired through the process of evolution. They're critical for embryogenesis and are silenced in terminally differentiated cells. An endogenous retrovirus, HERV-K/HML2, can get activated at specific loci in a subset of patients with ALS. The envelope protein can cause degeneration of motor neurons. Antiretroviral drugs can partially block the virus, and antisense molecules and shRNA can silence the virus and may have therapeutic potential. And so these are the people who actually do the work. As you can see, this is from the pre-COVID era where we could actually stand next to one another without our masks on. But this includes my lab as well as my clinical team here. And we work on endogenous retroviruses, HIV, and now in the last 2 years, our life has been taken over by coronaviruses. So we've been doing a lot of work on coronaviruses, but I have to acknowledge all of them for all the work that I've shown you here. And this is not possible without collaborators. And so I want to acknowledge all the wonderful collaborators who've been helping us for all these years in many different ways. And so I'll stop here and take any questions this time. Oops.
Thank you. Thank you so much. That's fantastic, Avi. Appreciate that. So there are a couple of questions. The first is from Al-Mudina. If HERV protein expression activation is upstream of TDP43 aggregation, what is activating in the first place?
Well, we don't know how these endogenous retroviruses get activated. I mean, Josh made a very good point that aging alone may be sufficient, but everybody who gets old doesn't develop ALS. So there's got to be more to it. So yeah, the inciting factor, I have no idea what it is. Maybe if you believe repeated excitation of a particular area can do it, then maybe sports-related people can get it. Maybe through repeated injuries. I don't know what initiates the process. There are a lot of potential hypotheses there.
I mean, is there I mean, presumably, anytime there is a significant illness, there is activation of retroviruses at some level.
No, retroviruses are very tightly regulated. If you activated retroviruses that easily, the human race would be extinct. Yeah, because they can cause cancer, they can cause degeneration. You got to regulate these things very carefully. There are very specific circumstances under which they will get activated and become pathological.
I guess what I'm asking is, if you have a broad range of pathologies that might, for example, cause infections, it might cause cytokine storm or severe.
No,
Yeah.
won't do it.
How often do you see HERV-K?
You won't get it. So we've tried all kinds of ways of stimulating it. We we put TNF, we put IL-6, we put NMDA agonists, on there. We put mitochondrial toxins on neurons. Never could activate it. We tried demethylating agents, SAHA and all that kind of stuff. Could not activate. You have to deacetylate and demethylate at the same time to activate HERV-K. It's not that easily activated.
Yeah, interesting. Thank you. Let me read a couple of other questions here. This one is from Nitesh Sangai. How does antiretroviral affect the proteinopathy in ALS? Do you want to mention through your research that patients who are on lifeboat ethics for drugs like edaravone should start antiretroviral drugs?
No.
What about the antiretroviral drugs which are known to cause oxidative stress? That's kind of a broad question, but
that's exactly the point. And one's got to be very careful. I can cite so many examples. So anti-TNF agents, where Cedric Rain had talked about how TNF is the cause of multiple sclerosis for decades. Anti-TNF agents work in rheumatoid arthritis. And then when we tried it in patients with multiple sclerosis, they have huge elevated levels of TNF. They all had relapses, right? So one's got to be very careful. Minocycline, the same story. I mean, you can see that people thought it was going to, what is it, just an antibiotic. I mean, people would do well, and ALS actually made them worse. So you got to do these things in proper clinical trials, because as I said, despite your best intentions, you can end up hurting people. And maybe whatever in vitro data I produce, whatever animal data we produce, we know things fail when you go to humans. Humans are very complex. So I would only do it in the context of a clinical trial.
And presumably a trial in which you profiled people for HERV-K before you treated them. So there have been negative trials, as you know better than I. For example, the BI in New York City conducted a trial with no idea at all of whether or not there were retroviral elements and concluded that retroviral therapy didn't work. So
that's the problem, because you don't know what you're treating when you do that kind of stuff. And I think this is the Indenovir that they used. And Indenovir has absolutely no effect on HERV-K.
Thank you. So Professor Dubnow asks, do you think that envelope transgene in mice requires the mouse endogenous elements to be expressed in order for it to be toxic?
Okay, so that's a good point, but we never looked at the mouse endogenous retroviruses, what's happening in this animal model, and probably should be looked at. But what we do know is we express only the envelope in the neurons in order to do it. And only the motor neurons degenerated, while the envelope is expressed in all neurons, right? Throughout the brain. So there's something special about these motor neurons that are degenerating.
Let me ask you this. Joseph Klim writes, are there other intersections between other Mendelian ALS genes and events that you identified as being signaled or important for HERV-K? In other words, you focused, and this is also for Professor Dubnow, but you focus a lot on TDP-43, understandably. But what about intersections with some of the other ALS genes?
I think they probably can intersect. Now, if you looked at the work that Molly and Josh did for transport, they say that all ALS is not the same, right? So it's like saying ALS in some ways like fever. You got multiple causes of it. And so it's quite possible there are subtypes that clinically look like ALS, but pathophysiologically they're totally different. And certainly the SOD1 ones look very, very different than these others. What we have done is we've looked at C9 open reading frame IPS-derived neurons, and there we do find activation of HERV-K. in those, at least in culture. So, but we haven't had a chance to look at all the autopsy tissue and stuff from those patients yet.
That's interesting. Thank you.
If I could chime in just for 1 minute on that same topic. In that paper, the Tamidal paper,
Yeah.
what Molly really noted is that there's these 3 categories based on expression profile, but it's impossible to distinguish 3 subtypes versus 3 stages, for example,
Yes.
because it's just endpoint, right? And you're looking at a chunk of cortex. So it's very hard to know, or it could be a combination of those as well.
Except the thing to remember is ALS, you can make that theoretical argument, but most patients with ALS, it's so short-lived. I mean, the time you develop the disease, the time you die, 50% in 3 years, 90% in 5. So, there's not much of a course there. And most people die at terminal stages of the disease. They're not dying very early in the course of the illness. So and from that standpoint, you probably are pretty homogeneous.
Raul Kraus asks, can neutralizing antibodies against the envelope protein be detected? And are they protective?
So that's the implication of our findings. We certainly haven't done a neutralization assay that typically like virologists would do. But what you can make an argument is that you could use a neutralizing antibody for a potential clinical trial if you wanted to see if it affects the course.
Thank you. And another question is asked by Jesse Tordoff. How far does the HERV-K particle travel through tissue? Can it enter the circulatory system?
So, I mean, once it's, for number one, I cannot say it's a particle yet, right? Because all I can say is Josh pointed out that these are immature particles, or they could be exosomes, or, they can come out of it. But once it's in the extracellular fluid, it can travel anywhere. There's no hindrance to it.
Mm-hmm. You and to that point, you showed pretty impressive data using CSF, including Western blots. Can you detect signal as well in the circulation outside the CSF?
We did all the HERV-K viral load that I was showing you, which we called HERV-K ratio, was all in serum samples.
Yep, that's all serum. Yeah. So, and I guess the other question I would ask you, and it may be premature, but this gets back to the question of other ALS genes. Just in the broadest sense, if you take a collection of CSFs from a whole bunch of Mendelian types of familial ALS, do you see differences in levels, or is this being done of HERV-K?
If you can give them to us, we'll do it for you.
Okay, so that's
the assay now is working beautifully in our lab. It took us many years to perfect the assay. Now it's working beautifully.
Yeah, I mean, it seems to me that would be,
and the ALS Association was very kind to give us some money to do the assay. So we've got it working well.
Yeah.
So if you want any sample, we can do it blinded. We'll give you the data and you analyze it and do whatever. It's no problem.
That's very interesting. Yeah. So in terms of Koch's postulates, you're almost all the way there. When you make a transgenic mouse that expresses high levels of the envelope protein, you see it in motor neurons and you get motor neuron degeneration. And I guess, is that the same as proving that it's HERV-K itself? Or is there a sense that there could be toxicity from high levels of envelope protein independently of the reality of what it does in disease? I'm pushing back a little bit mainly to get your comment on that, Avi.
Yeah. No, you're absolutely right. I don't think we've proven anything really. I mean, all we've shown you that yes, it plays a role in pathophysiology of the disease.
Yeah.
We haven't shown that it's the etiology of the disease. And proving etiology is very difficult. I mean, if you look at how much time did it take to prove HIV causes AIDS? I mean, it took over a decade or longer, right? It's not simple. So I'm not that naive to think that we've proven any kind of etiological association. All we can say is that it plays a role in the pathophysiology of the disease. And it's quite possible that maybe we are overexpressing the protein and we are showing that you put a lot of protein. But look at it this way. And we've done the same thing for HIV. We showed TAT protein was toxic. So people say, well, do you think TAT is the cause of HIV dementia? Of course not. But if you have dysregulation of glutamate, you got oxidative stress going on at the same time, and you add this protein on top of it, well, 1 and 1 doesn't make 2 any longer. 1 and 1 makes 3. So, but if you can block that take one out of the equation, you can actually slow that progression of that disease.
Yeah.
Right.
Yeah.
So it's the same way. I mean, I don't think anybody believes that, oxidative stress is the cause of all of ALS, but yet if you block oxidative stress, you can slow down some of the disease progression.
Yeah, absolutely. Well, this has been a fascinating session. And my own sense is that, among other things, as you both have said, an appropriate trial, designed and executed the appropriate way, is a crucial next step in this. And that actually brings us to a transition point, which is that in about 5 minutes, we will turn moderation position over to James Berry, who's going to start us on discussions of trials, trial design, and trial execution in ALS.